Welcome back. The next experiment is indeed an experiment by me. I did this experiment. Um, it's about preparation of a temporary stained squash of a grasshopper testis for observation of different stages of meiosis. This is the aim of the experiment. Quite an elongated aim, so that let me simplify. In the testis of grasshopper, we try to study different stages of meiosis. Because testis contains germinal cells and meiosis takes place in germinal cells, we chose the testis as an ideal material for the study of meiosis. That too, we chose the testis of a specific species of grasshopper, Poecilocerus pictus. The green colored grasshoppers, large grasshoppers, feeding on the leaves of a plant, Poecilocerus pictus. The beauty of this grasshopper is the presence of 19 number of chromosomes in the primary spermatocyte, male germinal cell, the male primordial germ cell, 19 number of chromosomes. If someone asks me about why 19, what about the number of chromosomes must be deployed at 2n number? Why 19? It's not deployed, it's an odd number. The answer is there is no Y chromosome. This is the mechanism of sex determination. One X determines the maleness and 2X double X determine the femaleness. So in the male grasshopper testis cells, germinal cells, there will be 19 number of chromosomes. And the second beauty of this is that all the chromosomes are acrocentric. Acrocentric means there will be 19 chromosomes and all the chromosomes will be acrocentric chromosomes. The centromere will be at an end. There will be two arms, but one arm will be very insignificant, the other arm insignificantly small, and the other arm will be quite large, so that it seems as though the centromere is situated at one end of the chromosome and there is a single arm. No, there are two arms, but one arm is so significantly small that it's difficult to locate with the help of a low power microscope. Then what is done? The sexually mature male grasshopper is taken for dissection. What we do? We dissect out the testes and then we put the testes in an insect saline solution. We clean the testes of the fat bodies and then we transfer this testis, cleaned testis follicles. Testis will be made up of several follicles. We then transfer the testis follicles into a fixative. The fixative's name is Carnoy's fluid. Carnoy, C-A-R-N-O-Y, Carnoy's fluid. Sorry. Carnoy's fluid is also known as aceto-alcohol fixative. In aceto-alcohol, there will be 3 parts of absolute alcohol and 1 part of 45% acetic acid or glacial acetic acid. Both are mixed thoroughly and used as a Carnoy's fluid or aceto-alcohol fluid for the fixation of the testis follicles. The testis follicles may be put for an indefinite period in the aceto alcohol. Whenever a preparation is required, one or two testis follicles are removed from the carnoid fluid and the preparation is gone through. So what is done? Next, that we have had the testis follicles fixed in a carnoid fluid. Next, we take a slide, microslide. Then put here, just in the middle, 1 or 2 drops of glacial acetic acid. Sorry, not glacial acetic acid. I'm sorry. I withdraw my words. Then the testis follicles are removed from the fixative and put in a cavity block. There is a block of glass wherein there is a cavity at the center. This is called a cavity block. We must stain it. We use carmine stain or Orsin stain. Remember, carmine or Orsin. Usually, the usual practice is carmine stain is integrated into the fixative aceto alcohol. The resultant solution becomes acetocarmine solution. There is no need, but the usual practice is this. What is done? Aceto alcohol is prepared and to it is added a recommended quantity of carmine powder or Orsin powder. It becomes acetocarmine. We usually follow whatever our forefathers or predecessors have said and done. But in my opinion, when the tissue is fixed, the testis is already fixed, there is no point in using acetocarmine. Only carmine stain solution will work out the needful. Why do we use again the alcohol? Anyway, so carmine solution or Orsin solution. We have taken the testis follicles in a cavity block. Then we are adding some carmine stain solution or Orsin stain solution. We wait for 30 minutes or so. 30 minutes. Allow the testis follicles to stain for 30 minutes. Then remove the stains of testis follicles from the stain solution. Now it is ready for a squash preparation. In the squash preparation, we take a clean microslide. At the center of the microslide, we put a drop or two of glacial acetic acid, that is 45% acetic acid, and then we transfer 2 testis follicles. Remember, more testis follicles will create more problems. There will be overlapping of cells. Take 2 testis follicles, put on the glacial acetic acid. Over it, you place a cover glass, thin cover glass. Okay. Between the two, put the slide in between 2 folds of a blotting paper. You take a blotting paper, you hold it like this and place the slide here. You take a blotting paper, you fold it like this. Okay, here is the blotting paper. You take a blotting paper, hold it, and in between the 2 folds, you place the slide like this. You have put a cover testis follicle in glacial acetic acid, 1 to 2 drops. A cover glass is mounted. Then you put a blotting paper, fold. Then you place it like this. Just hold it in a clockwise direction. or counterclockwise direction, whichever is convenient, never in both directions. In a clockwise direction or counterclockwise direction, you press it gently, not so hard, gently, in a gentle manner, you just press. What happens? The stem testis follicles that contains germinal cells in different stages of maturation will burst, the testis follicle will burst and these germinal cells will spread on this slide. Will spread on this slide. Then what is done? Nail polish is added, is rubbed at the edge of the test, cover glass. Nail polish. The nail polish that he used to paint the nail, that is the substance. That is added here at the edge of the cover glass, so as to prevent the entry of any outside air into the preparation. If outside air enters into the preparation, then everything will undergo decomposition, microbial decomposition. So to study, to ensure that you need to study today or tomorrow or a day after, you seal it with nail polish. There is no guarantee that this slide, this is a temporary preparation. Mind it, this is a temporary preparation. It doesn't ensure you to preserve it for the posterity. You will prepare it, you will study it, then you will discard it. But for 2 to 3 days preservation, you apply nail polish. Avoid the entry of air into the chromosome squash area. That's it. So this is how we prepare a squash preparation. It is now ready for microscopic observation. A microscope, a high resolution microscope, is used for studying different stages of meiosis. There are 3 magnifications. One is 10 into 10, other is 10 into 40, another is 10 into 100 magnification. These are the 3 usual magnifications in a moderately good, not very good, moderately good compound microscope. These are the 3 magnifications available. You need to scan the slide at 10 into 10 first. 10 into 10, you locate the cells, then you go for 10 into 40 magnification. Then if it is not resolved, you are unable to resolve the things, go for 10 into 100. But for higher secondary, for you students, 10 into 100 magnification is not the desired magnification. You may not be able to handle 10 into 100 magnification. Just hear about this, but don't try to practice. Only 10 into 10, 10 into 40. You practice. So after scanning, we got these following stages. Prior to going into the details of the stages marked there, let me tell you a little about meiosis. Meiosis is a reductional division. Mitosis is an equational division. Reductional division means the number of chromosomes present in the parent cell are reduced by half in the daughter cells. Equational division means the equation is maintained between the parental cell. Whatever number of chromosomes the parental cell contains is maintained in the daughter cells. There is an equation, equal number of chromosomes. So meiosis, we are studying about meiosis. Meiosis consists of 2 successive divisions. One is meiosis 1, the other is meiosis 2. Meiosis 1 indeed is a reductional division wherein the number of parental chromosomes is reduced by half. Meiosis 2, the consequence is the formation of, if the parental cell is diploid, the daughter cells will have n number of chromosomes, haploid, 2 haploid cells will result at the end of meiosis 1. Meiosis 2 will follow immediately. No transit time is there. Meiosis 2 is equational. It is equivalent to mitosis. The number of chromosomes in the parent cell is retained in the daughter cells. So at the end of meiosis 2, there will be 4 number of chromosomes, each containing n number of or haploid number of chromosomes. 4 number of cells, each containing haploid number of chromosomes. Well, meiosis 1 is very important because how the number of chromosomes are reduced by half. We have placed our focus mainly on meiosis 1. Meiosis 2 is of little importance. Anyway, we have also studied meiosis 2, but more focus is laid on meiosis 1. Meiosis 1 consists of the usual phases of a cell division: prophase 1, metaphase 1, anaphase 1, and telophase 1. Similarly, meiosis 2 will have the usual phases. Add the suffix 2, prophase 2, metaphase 2, anaphase 2, and telophase 2, meiosis 2. These are some of the stages, important stages I have chosen to bring to your notice. Please make a closer focus. Look at it. This is, of course, let me tell you prior to that, let me tell you, I have noted down everything, but haven't told you. Meiosis 1, prophase 1, I'm sorry. Prophase 1 is by far the longest phase of meiosis. It is again subdivided into 5 subphases, namely leptotin, zygotin, pakitin, diplotin, and diakinases. These are the 5 sub-stages of stage prophase 1. Each stage has its characteristic features. Based on these characteristic features of chromosomes, the names have been coined. So leptotin, leptos, thin, tin, thread. So thin thread stage. Zygotin, pairing stage. Pakitin, chiasma stage. Diplotin, crossing over stage. Diakinases, terminalization stage. These are the stages, 5 stages. Okay. In leptotin stage, the chromosomes are very extended. Chromosomes are very delicate and thin thread-like structures wound around each other, forming a mesh. So it is very difficult to identify an individual chromosome during the leptotin. In zygotin, we have skipped over it because any cell can be designated, any cell having a nuclear membrane can be designated as being in leptotin stage. We have skipped over it. Then zygotin, pairing stage. The same situation will be there. It's very difficult to distinguish the chromosomes. There will be bivalence pairing stage. The maternal chromosome and paternal chromosome, they will undergo pairing. Bivalence will be formed. Pairing stage, in the pairing stage, but it's difficult to identify. Towards the end of the zygotin, you will have some kind of a visibility. Then comes the pakitin. Pakitin is the chiasma formation stage. Look at this diplotin stage. This is only diplotin stage. These are 2 chromosomes. You can see very well. This is a clear sight, clear picture, clear sight. Please focus on this. Look at one of these elements. Look at this element. You can see 2 elements. They have come close to each other. This is one. These 2 elements are known as 2 chromosomes. Each chromosome now has 2 chromatids, sister chromatids. So the 2 chromosomes in all will have 4 chromatids. The stage is known as a tetrad stage. Here, see, at some places they are apart from each other. At some places they are very closely associated with each other. The point at which the chromosomes are closely associated are called chiasma. Look at this chiasma. Chiasmata is plural form, chiasma is singular. This is one chiasma, these are chiasmata. Okay. So this is called. I'm referring to the Pakitin, Lake Pakitin. From the Lake Pakitin, through a transition, the cell enters into a diplotin stage. In the diplotin stage, there will be crossing over. Here, crossing over is taking place. See, exchange of homologous segments between non-sister chromatids refers to crossing over. Exchange of homologous segments between non-sister chromatids of the two parental chromosomes is known as crossing over. Crossing over leads to genetic recombination. This gives rise to variability. Okay, this is diplotin and this is diakinesis. In diakinesis, what happens? These chiasmata move towards one end, one of the poles. These are two poles, towards one pole. The chiasmata will move. This movement of the chiasmata is known as terminalization. Unless they move to the terminus, they will not separate from each other. So terminalization here, look at this. Terminalization has taken place. This is most condensed here and terminalization has taken place. This is a low resolution photomicrograph, so the resolution is not as clear as in the previous slide. Next, this is diakinesis, terminalization state, prophase is over. Next comes metaphase 1. In metaphase 1, the chromosome bivalents are arranged along the equatorial plane. So along one plane, equatorial plane, look at this. If this is the cell, this is one plane, all the bivalents are arranged along the equatorial plane. You can count the number of bivalents. Can you tell me how many bivalents will be there? If there are 19 chromosomes, there will be 9 plus 1, 10 bivalents. 9 autosomal bivalents and 1 X chromosome. Only this much. So you can count the number 1, 2, 3, 4, 5, 6, 7, 8, 9. One of these is an artifact. This is an artifact, not a chromosome. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 9 plus 1. 9 autosomal bivalents and 1 X. This is a clear case of metaphase 1. Then comes anaphase 1. This is anaphase 1. Look at the separated chromosomes. The chromosomes, how many chromosomes will move towards each pole? Towards this pole, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 and 11, this is of a different grasshopper. No. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 this side and 9 this side. There will be, this is an artifact. This is an artifact. So discarding the artifacts, we count the number as 9, 10 and 9. 10 to one pole, 9 to other pole. In all there were 19. 10, 9. So here the number of chromosomes is reduced by half. You may ask me 19. It will become half. 9 half. It is practically impossible. 9 half. Half of one chromosome. Either 9 or 10. Or 10 and 9. 10, 9 or 10, 9. Like this. So anaphase 1. In anaphase 1, the moving chromosomes look V-shaped. V-shaped. Look at the V-shapes. This is metaphase 2. In metaphase 2, the chromosomes are V-shaped and are arranged along the equatorial plane. So they are arranged along the equatorial plane. In anaphase 2, the centromere holding the two chromosomes together break in the middle and the chromosomes acquire rod septs. They become rod sept chromosomes. Here, look, there are two cells lying side by side. This is one cell, this is another cell. Anaphase stage, this two is in anaphase stage. The chromosome groups have started moving to the opposite poles. So, how many chromosomes? This will have 9, this will have 9, this will have 10, this will have 10. Or 10, 10, 9, 9. In all 19 chromosomes. You add it up anyway, it becomes 19. So, division. So, if cell has, for example, if a cell has 19 chromosomes, that is plus X. It will divide. In the first near division, the number is reduced. 9 plus X. 9 plus 0 or nothing. You don't like 0, 9. Okay. In the next division, 9 plus X. This is equation. 9 plus X. 9 plus X. And this is 9. Equation. This is meiosis 1. This is all about meiosis. But the thing is that it's a very long procedure. You may not complete the procedure within 3 hours, 2 hours, or 1 or 2 hours. Therefore, you will be asked to identify a stage. My objective of telling you all this is to go through the shapes of chromosomes, how to identify. You can, looking at the chromosomes only, you will not be asked to prepare a micro slide. You will be asked to identify a stage, meiosis, stage of meiosis. Looking at the chromosomes, this gives, if you have a fundamental concept on chromosomes, the chromosomes I have already mentioned are apocentric, and 19 number of chromosomes are there. If looking at these figures, you can see which figure is in which stage. So that's all about from me. Thank you very much for giving me this. Thank you very much once again.